Hello everyone, my name is Wilson and I will be presenting over how genetic background determines the severity of age-dependent cardiac structural abnormalities and arrhythmia susceptibility in SCN5A-1798 INS capital D mice. I know that was an earful, but bear with me and I hope you enjoy that little ratatouille photo. So, with the whole point of the study, you have to kind of need to understand the anatomy and how your heart works. To just give you a general baseline, your heart has four different chambers, okay? It has the atrium and the ventricles, both left and right, left and right. So there's four. They all kind of work in tandem to pump in and out oxygenated and deoxygenated blood. But what begins or what helps out with the contraction of the heart muscle is going to be the electrical signals that help conduct this type of action, which is why this whole system is called the cardiac conduction system. So in anatomy, the first thing that you always learn is how the SA node is the pacemaker. The pacemaker being this is the part of the heart that first generates that first electrical signal, the electrical action potential, okay? This is where your atrium contracts, and then that signal then goes to your internodal pathways, then to your AV node, then to your bundle of His, then right left bundle branches, and then Purkinje fibers. Where, as you can see in the Purkinje fibers, it kind of encapsulates all of your your ventricles, your left and right ventricles, which is where the Purkinje fiber is responsible for the ventricular contraction as well. So why is any of this significant towards our study is because the gene that we're looking at is the SCN5A gene. This gene is responsible for the sodium voltage gated ion channels that that gene specifically encodes the protein for the alpha subunit for these ion channels. Basically, what these ion channels do is it helps the smooth transfer of the signals from the SA node all the way down to the Purkinje fibers through the opening and closing of these sodium channels. Okay. The analogy that I found pretty useful to kind of explain this to people that might not know much about how the heart works or what I'm even talking about at all is this gene with these sodium gated voltage gated sodium channels allow for the like kind of like the how dominoes fall, right? It kind of goes all in a smooth straight line when you tip one over and then just kind of din 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 din. So it's the same thing with the voltage gated sodium channels. So why is this even important? A lot of the gene mutation from the SEN5A can be tied to heart conditions like Brugada syndrome, long QT syndrome type 3, sinus node dysfunction, bradycardia, cardiac conduction disease. During the study, I also found that there were ventricular arrhythmias or sudden cardiac arrests in young individuals that are healthy, if not healthier than the average individual. And for all the syndromes and diseases, they're all just heart conditions that are either giving an extra beat or beating too slow or beating too fast when talking about when your heart contracts and rests and everything. So the aim of this experiment was to figure out if this mutated gene um, has other modulating factors like genetic background or the age. So how they did this study was they took two different strains, which is the FVBN and then the 129P2 strain, and compared it to each other, as well as the adult versus the age or the elder version of these mice. So the adult ranging from 2 to 7 months, and then the age being 8 to 28 months. And then lastly, obviously the mutation was the main emphasis. So you'll have your wild type of those strains, and then the mutated type of those strains. The methods that were used for this was the in vivo electrocardiogram, basically a heart reading, which you will see later. In vivo meaning while the organism was alive. And the whole heart electrophysiology, basically the understanding and study of your heart and how the conduction system and everything works. Echocardiography, basically an echo. If you know what an echo is, it's just, there's this machine that makes sound waves and produces like an image for you to see. And then the fibrosis quantification, we'll get into that later. But mainly they use staining to figure out what parts of the heart was developing like scar tissue. Lastly, statistical analysis. There's a lot of different methods, techniques they use. Three-way ANOVA, Shapiro-Wilk normality test, going into a non-parametric Crucible-Wallace test, and then if there's any statistical significance, they use a post hoc analysis. So to further to interpret all of this data, all of our results, the main takeaway, I always wrote it in a red arrow, would be how in here you can see the aged mutants were the most affected, but in general, this whole strain 129P2 had more ventricular extrasystole, ventricular tachycardia, just a lot more heart problems and heart conditions. The EKG or echocardiogram that I was referencing earlier, this is what that reading um, is here for. No need to further interpret that. And then in this slide is interpreting the gram, like the whole echocardiogram. But the main takeaway on this slide is how the PR interval, the QT duration, the P duration, and the QRS duration, as well as the QTC duration, are all affected mainly by age. But as you can also see, there were big differences on the strain of 129P2, comparing this to the FVBN. And if you're confused by the colors, you can pause the video and look at the key up here on top to see the main differences. Let's see. Here, this is depicting on a bar graph, the delay between the AV and the HV, AV being from the atrium to the ventricle, and then HV being the bundle of His to the ventricle. There wasn't much of a difference in the AV delay, though there was a subtle, which I'll show you, like a subtle difference at the end, which I'll show you in the next slide. But from this, I wanted to mainly point out how over here you can see there's differences in the mutation and the wild type of the 129P2 strain. I use this red arrow to point out the how shorter coupling intervals on the AV delay for the adult and aged of the mutants between both strains were the most noticeable. And then here you can also see the left ventricular pacing as well as the right ventricular pacing. Basically, they put leads or leads onto the parts of the heart that they were testing, and they found a lot more problems in the right ventricle than the left ventricle on the mice. Mainly being arrhythmia in the sense is indicating any incidents of arrhythmia that might have happened during the test trial that they were monitoring. I had a red arrow right here because you can see a big difference between the strains as there's an increase in the mutants, but especially in the aged mutants on the 129P2 strain. And then over here is the ejection fraction. That's basically how efficient your heart pumps out blood. The 129P2 mutant strain is very obvious that it's not as efficient um, as the wild type, and there's not much of a difference here. And then the RV size is your right ventricle size. So they can also label this as ballooning if there's an enlargement of the right ventricle. So as you can see, the aged mutants of the 129P2 strain exhibited a lot more of this change. This is demonstrated through the pictures. You can see in the 129P2 age mutant strain, there is an enlargement right here that's kind of outlined in the yellow. And then on the right side where the F part is, you can definitely see where the heart is right here and how that's enlarged compared to the FVBN strain. Both mutants. And then here is the cystic fibrosis portion of the experiment that they did. These were paraformaldehyde fixed hearts that were stained with picrocereus red stain. Um, there's obviously some that you can see in the FVBN from the wild type compared to the mutant type, but there is an obvious difference from the wild type compared to the mutant type in the 129P2 strain. And this right here is showing just immense amounts of cardiac fibrosis. So just lots of scar tissue, it can affect your just everything within your heart, whether it's the efficiency of how it contracts or not letting your heart relax a certain way. So all in all, with everything, cardiac electrophysiological and structural abnormalities are heavily affected by genetic background. As we saw, there was a lot of differences in the 129P2, as well as how the aged groups were more affected than the adult groups. Within looking at this SEN5A gene mutant version, um, the good thing about this study is it kind of gives us an indication of what to look out for in terms of what type of population we might want to test more for at risk for arrhythmia, as well as just other clinical therapeutic studies that we could do to try to help patients with these conditions. The weaknesses that I do want to point out in the study is that the sample size wasn't the biggest, and also when I meant by limited genetic backgrounds and species differences, first off, genetically, we had to use a mouse model or a mice model, which is a animal model because you can't necessarily do it on humans because of all the other cofactors like diet or everything else that you can't control, and it's small sample size amongst humans. Um, and then but yeah, there's just a big genetical difference between mice and humans, but it allows us to have kind of a idea of what certain things could be or how this might affect one thing, right? Specifically the SCN5A mutation. But lastly, the last weakness I want to talk about was the only one mutation, which we're, only looking at SCN5A. What if there are other type of genes that affect arrhythmia? But yes, thank you for listening, and I hope you have a great day.